to Nutrition Rounds with Dr. Danielle Belardo, the podcast for anyone interested in learning about plant-based nutrition through an evidence-based approach. Every week, we share insights and interviews with physicians who are leading experts in nutrition and health. Whether you've been plant-based for many years or are still searching for the perfect diet, Nutrition Rounds will inspire and empower you to live your healthiest life backed by science. Now here's your host, Dr. Danielle Belardo, MD.
All right, in today's episode, we are with Dr. Tang from Cleveland Clinic. I am beyond honored to have Dr. Tang on the podcast. He is absolutely brilliant, and I cannot appreciate more for Dr. Tang taking the time out of his busy schedule to talk with us about TMAO and all of the research he's done. So thank you, Dr. Tang, for joining us.
Oh, thank you, Danielle. It's a pleasure to be here.
Thank you. So first, I would love for you to just introduce yourself and tell everyone kind of about what you do and what your work as a cardiologist and a researcher.
Well, I'm actually a heart failure transplant cardiologist at the Cleveland Clinic. And my day job, I see patients with heart failure and trying to identify, potential causes and trying to delay the progression. And if, unfortunately, they get worse, I try to see what we could do with mechanical devices or transplants and then actually take care of them afterwards. So obviously, that's very different from my research work, which is trying to prevent that from happening. It is too many people are heading towards the same cliff, and we definitely need to find ways to prevent them from developing heart disease and, more importantly, heart failure.
Absolutely. And your research is just outstanding. Well, everything you do is incredible. Heart failure and transplant is obviously a very amazing aspect of cardiology because so much is changing with our different, not only medical therapies, but now with transplant and some of the mechanical circulatory support. So it's really a dynamic, changing, and unbelievable field. But your research is so groundbreaking and fascinating to me. And so I would love if you could give everyone, there's a lot of physicians, scientists, and science lovers who listen to my podcast, and a lot of them are interested in the work you do. So I'd love if you could kind of give an overview, just starting kind of about what TMAO is and your work and how it relates to cardiovascular disease.
Sure. So I've always been fascinated by how the heart interacts with other organs, and in particular the kidneys. And so for a long time, I've been working with different researchers to look at how the heart and the kidney interacts. And my translational research has actually headed towards that direction early on when we were trying to find molecules in the blood that distinguish between those who progress and develop, cardiovascular events versus those who do not. And early on, I measured a lot of small molecules that we actually known to have cardiovascular risk, something like the ADMA, asymmetric dimethylarginine, which is really an endogenous nitric oxide synthase inhibitor, and also different small molecule metabolites. So that's actually where I started looking at oxidative stress and nitritive stress and how, using blood and urine, we could actually detect kind of early triggers that can help us identify, progressive diseases. So we have this kind of idea that potentially there are many unknown substances in the blood that also tell the same story. And when we first started, we have a mass spec machine that could identify different small molecules. And we have samples of patients who subsequently developed diseases that we have collected early on with a control group of those who did not develop disease. So we actually tried over and over again using different case control comparisons to look at a vast array of molecules, small molecules, usually under like 100 Daltons, molecular weight of 100 Daltons, and trying to figure out, these small molecules, whether they could distinguish patients who have events versus don't have events. And as you imagine, when we keep going this case control, comparison over and over again using different, cohorts of samples, we ended up sieving down or filtering down less and less number of molecules until we hit about 18 of them only after like multiple successions of case control comparisons. And so that actually seeps out from thousands of molecules to about 18. But then they are all kind of in molecular weight. So we actually don't know what they are. We knew one or two of them, but we didn't know the rest. And so it's a hard work of a few postdoctoral fellows from Dr. Stan Hazen's lab that started working on literally figuring out the permutation and combination of these potential compounds. And then, of course, we have to validate them. As you can imagine, many, many different small molecules have the same molecular weight. So if you think about it, we are literally doing try and error at that point. That was kind of a hectic work. But then we certainly find 3 molecules in these 18 that seem to be correlating to each other, and we were very perplexed as what they are, and so we actually focus our efforts in looking at that, and it turns out they are all having a trimethyl group, so 3 methyl that link together, and one of them seemed to be a byproduct that is a conversion by humans, but it was generated by bacteria. In the literature. So that's how we actually, accidentally got into the whole field of gut microbiome. Obviously, never predicted that this is going to happen, but we started to be able to, specifically identify TMAO, which is trimethyl and trimethyl and oxide, trimethylamine and oxide. So this is actually how we actually identified that. And so after that, we have, obviously other samples that had long-term outcomes, and we started to kind of generate kind of the prospective cohort of patients who we know over time had, a certain number of people have diseases. And we started to, analyze those samples and independently identify the prognostic value of TMAO in those cohorts, which is independent of all the traditional risk factors. And to demonstrate that indeed it may be a causative factor, we have to turn to animal models. And conveniently, the mouse model that we had the bacteria in the gut that produces a good amount of TMA that convert to TMAO. We kind of learned about that subsequently because some mouse actually don't have those bacteria that makes TMA, which becomes very convenient for us otherwise. But the most common laboratory mouse was the black 6 mouse, and in fact, it does generate a lot of TMAO when they were fed with choline diet. TMA really comes from bacteria consumption of substrates the food that we eat every day. So anything with methyl groups that they could actually use as food, the bacteria use as food, ended up generating this nitrogenous byproduct called TMA, which it goes into the blood and through the liver, and the liver actually converted to TMAO.
So the foods that are most high in choline and carnitine, which are the precursors when they go through the gut microbiome to go from TMAA to TMAO. So these you find highest in, you would say, things like eggs and red meat, things like that.
Yeah, meat, liver, things that actually have high cholesterol. It's kind of, some people even call it the third cholesterol. And this is something that all the things that we as cardiologists tell our patients to avoid for a good balanced diet have high choline content. Now, just be aware that choline is important. Some people feel quite strongly that in early development, we do need choline to it's a precursor for acetylcholine, which obviously is important for neurodevelopment. So that's why it's actually advocated as a supplement for pregnant women. And there are actually consumption guidelines for that. And some older patients also use choline for that purpose, thinking that it may actually help with reducing, cognitive decline, except that obviously the data there is not very strong. But I think it is important to recognize, and this is where we found out later, and probably interesting to your audience, that another compound that is common in food groups is carnitine, which is in meat, in red meat in particular. And that certainly has a different type of enzymes and different types of bacteria that convert the carnitine into TMA and subsequently lead to production of TMAO. So I think that is where we have the unique opportunity to truly understand what the environment, in this case, the dietary intake in different food groups, in particular meat, and how that impact the body's response and also the metabolites it's produced when eating red meat versus not eating red meat.
That's fascinating. So when did you start to make the connection between TMAO and atherogenesis, platelet aggregation, atherosclerosis, things like this?
Yeah, this is actually quite early. Once we actually got the animal models going and recognized that the mouse that we use indeed when eating choline, phosphatidylcholine in particular, lead to production of TMA and TMAO. Obviously, we were able to start using the mouse model to look at atherogenesis. So what we do is we were able to feed the mouse with or without the choline content in the chow. And then this is the type of mouse, the black 6 mouse, that are more prone to development of atheroma. This is the so-called apoE mice. And so studies have actually shown that with that mouse, they are like, accelerated atherosclerosis. And interestingly, when we were, we were able to demonstrate, firstly, the bacteria is involved in this by giving both antibiotics and see whether the TMAO levels go down, which they did, but also for germ-free mice, when they don't have any gut flora, when we give them choline diet, they don't generate TMAO or TMA and TMAO, but then when the mouse was exposed to kind of, the wild, meaning they start accumulating gut bacteria, the TMAO production increases. And the presence of this TMAO elevation leads to more atheroma in the aorta. And when we give a short-term antibiotic with a triple regimen, something that stays in the bowel for the mice, we actually found that is attenuated. Wow. So all these really had a direct causative effect, because now we could demonstrate, almost like the cause postulate where we learn in infectious disease, in microbiology, that the presence of the bacteria is essential in making the metabolite. And obviously, TMAO is also present in some food, particularly fish and seafood.
This is 100% what I wanted to ask you. This is like the number one question I wanted to ask you is consuming preformed TMAO in fish, does this raise serum levels in humans? And if so, is a rise from that form of TMAO bad for us?
It does. In fact, if you actually have, just otherwise healthy individuals eat a wide variety of food, which people have done experiments on that, the main food component that leads to a transient rise and fall in TMAO is fish. And but not all fish has the TMAO. You think teleologically, TMAO is made by the muscles of the fish, particularly deep sea fish, because it changes the melting, the freezing point of the muscles. Is actually the muscles anti-freeze. So it provides a protection for the deep sea fish. So that's why the deep sea fish doesn't look like the fish in the freezer. It actually makes the fish not freeze. And so in fact, people have done, there's a lot of work in marine biology on this. And in fact, there are enzymes that are made in the fish's muscles that actually convert TMAO as well, or generate and also eliminate, because when fish, moves from colder to warmer waters, actually some of this also change. So we actually know that not all fish is created equal too, not only from TMAO levels, but also their fish oil levels. So if you actually measure TMAO levels in, say, lake fish or, shallow water fish, they actually TMAO levels are quite low. In fact, some of them are, very, very low concentrations. Whereas deep sea fish like cod and fish that we actually, tuna, or sea bass, they actually have much higher levels of TMAO levels. So in some ways, it is important to recognize that, not all fish has, high TMAO levels. And it makes sense that because of the function of what it does, that deep sea fish, which is also the type of fish that is related to baked fish or fried fish, has higher levels of TMAO.
Wow, that is actually really interesting. Yeah, that was a question I've been dying to ask you for quite some time. And actually, I can't even take full credit for that question because before our interview, Rob said, I want you to ask. Rob Ostfeld was like, ask Tang this question. And I was like, everyone wants the answer to.
Yeah, in fact, we actually, our lab actually did work on trying to measure the different, other people have done that. In fact, the very interesting thing about TMAO, there's a huge amount of literature in the sewer literature, in basically waste, disposal literature. And partly because TMA is a really pungent gas, and TMAO is a byproduct. So in the early days, there was a lot of interest to better understand how to eliminate bacterial production of waste products. And so there's a lot of work in the sewer industry on TMAO and to understand how different metabolites of bacteria is generated and their source of generation. And in fact, all this work has been, done. It's just that we are, transferring this information inside our body. One can even conceive that, the body's main job is to detoxify all the things that is occurring.
Right. The scent of TMAO is that pungent smell that you smell in fish, right? Like that is.
Yes, in fact, yeah, it's kind of like a rotten fish smell. So, yeah. In fact, the main reason why our trash smells bad, over time, particularly in the summer now, is because there are many of these gases that are being produced by bacteria, which makes you wonder what is going on as we keep, our food as leftovers. And, it's not just TMAO, it's just like in general, the metabolism that's going on in bacteria occurs not only inside our body and where we reside, but also outside and everywhere else. So in some ways, this is a very complex ecosystem, both inside the body, but also outside. And food as it comes throughout the process gives you a pause in terms of understanding the importance of freshness and also the way you prepare it.
Absolutely. And so I have to tell you, Dr. Tang, I've read so much of your research, but I loved your JACC state-of-the-art review called Intestinal Microbiota and Cardiovascular Health and Disease. And for anyone listening that wants to read some of Dr. Tang's work, this paper in JACC this year is outstanding. And I love that. It's so great. I love that you discuss starting with a normal intestinal microbiome, and then you discuss dysbiosis in our microbiome with heart failure and dysbiosis in hypertension and diabetes. Do you mind just kind of describing what you discussed in the paper with regards to what happens in our gut microbiome? What do you see in our gut microbiome in people with these illnesses?
Yeah, we certainly is in this infancy of our research. And I think the advances that we have has been our ability to go beyond just culturing these bacteria. We've learned a lot now that only a subset of them could be cultured in a traditional way of microbiology. So by having sequencing, capabilities and also the dictionary to allow us to identify different, phyla, and then now get to the species level with shotgun sequencing. I also describe different methods that have evolved over time. We now are able to truly characterize, almost like, having a census of all the things that are living inside us. There's still a lot of limitations because our sample collection remains kind of what we, excrete. So we've known that different parts of the body truly, it's just like humans habitate in different places. Bacteria and phages and viruses also pick places in in the gut to stay and basically set up shop, and set up homes. And so, in fact, one thing that I've learned after doing all this investigation is the fact that most bacteria lives in the large bowel, in the large intestine, is because there's this very efficient recycling system through the bile duct. So the bile actually, when you actually absorb a lot of things, the bile actually try to, literally recycle a huge amount of these different substances, particularly choline. In fact, chole stands for bile. And so you can imagine anything distal from that, just like mosses next to a waterfall, the food is right there. So all the bacteria just stays there. And in particular, this is actually very important for us in terms of being healthy, because the bacteria, the majority of what they do is to digest our food and actually extract different, components and compounds so that we're healthy, whether it's vitamins or other cofactors. So, and the metabolites they generate that we've learned, for example, the short-chain fatty acids from the fibers, as well as the different small molecules that we are beginning to recognize and learn, are quite essential for everyday functioning. So one thing that people have been able to do is start to find individual groups of patients with different diseases and try to characterize, literally doing census on these populations, these bacterial populations. And right now we are still at its infancy, because all we can see is like certain, definitely for most chronic diseases, the diversity of the population is much lower. So the more bacteria variety is in more healthy individuals, and less variety is in, say, heart failure or renal failure and diabetes. And we have actually learned a lot about, in diabetes, there are specific interactions with different metabolites, by many other investigators on the branched-chain amino acids, as well as different other metabolites that are related to the insulin-resistant profile. In fact, I think the Israeli group has even suggested that the microbiome can predict an individual's ability to generate high sugar levels with different types of food. So that's actually something that we've actually have a lot more information based on very extensive investigations. So what we, however, have not recognized is how that translates into our clinical insights into individuals, meaning that the description of different compositions doesn't equate that we understand the function. We only do literally head counts of different types of bacteria, but not necessarily knowing exactly whether they're active, not active, whether they're generating metabolites or not. So this is the challenge, the ongoing challenge. And TMAO is really representing one prototype, should I say, of potentially many different types of metabolites that are being produced. And it's one of the more obvious, metabolite that, travels through a food substance that both the bacteria eat and we actually absorb, and how the bacteria and the human host interacts. And it almost become a marker of how things travel. But at the same time, it seems to have some mechanistic, relevance. For example, we have learned that those people who, both people and in animal models that have, elevated TMAO levels have a higher risk of clots being formed when the platelets are activated. And so that is another area of why, people have events. Remember the very beginning when we first started is to try and figure out who has events in the future versus no. Right. So one of the more susceptibility, and the reason why sometimes we think that, antiplatelet like aspirin is helpful, is because potentially, that gives you a little bit more vulnerability. It doesn't mean it causes it. It's just that if something triggers, you are more likely to actually have platelet activation, which is always in some ways bad.
So what is the actual mechanism, like the actual mechanism of the TMAO molecule, and how does it actually trigger activation, causing platelet aggregation and things like this?
Well, we actually still don't know. So I think Dr. Hazen's group actually tried to kind of work on this mechanism. We do know that the calcium cycling is altered. So there's some, intracellular mechanism that has activated. Obviously, the logical explanation is there may be a receptor somewhere, and we have tried to find it for the last, I don't know how many years, and we still have not found whether it's a service one or internal one. We can't tell because we have searched for it for a long time. One other thing is interesting. Obviously, you talk about aspirin, for example. One thing we actually published this work as a short research letter in Circulation about 2 or 3 years ago was we have individuals that were taking choline supplements, so they are like their TMAO levels actually were elevated because of that. And we then give them aspirin. And interestingly, yes, the platelet aggregation, potential is reduced with aspirin, which is expected. That's what aspirin does. But one thing that surprised us all was the actual TMAO level also reduces. The actual systemic measurement of TMAO actually drops, which makes us wonder because, aspirin, as you know, the current indication for aspirin is controversial. I mean, there has been some data suggesting that, well, not everybody should be on it. But right now, at least the preventive guidelines do suggest that aspirin should be used for people for prevention of colon cancer. And lo and behold, choline and all these, and even TMAO has been related to some risk of, subsequent development of cancer. And one may wonder, and this is all, hypothetical, we still don't know. We're actually doing studies trying to figure it out, is maybe some drugs that we use may modulate the ecosystem of the colon. Wow. And if that's the case, that may actually ended up changing the compositions or changing the functions of the bacteria that lives inside us that ended up making less TMAO. So if that's the case, that may explain why, for some people, particularly those with more elevated TMAO, may be more beneficial if they may be higher risk of adverse event, and in that case, they may be the identification of that is allows a subset of patients may be more beneficial for aspirin therapy, and that need to be prospectively tested. That certainly can explain the intersection between the different benefits of aspirin.
Yeah, that's fascinating. The first time, so the gut microbiome research has just blown up. It's just really evolved for the past several years. The first time I really even learned what the gut microbiome was, I was an intern at Temple, and I remember I was on the gastroenterology service, and we were talking about using fecal stool transplants for patients with C. And I thought it was fascinating when I found out that, you can actually do a stool transplant for anyone. The donor stool has to be coming from someone with a BMI less than 25. And the reason why was there was research that showed that when you had transplanted the feces, the stool, from a person that was actually obese into someone with C. diff that wasn't, they ended up gaining weight. And so I thought it was fascinating because it was like my first realization that, wow, these bacteria that live in our gut really do change things systemically. They're not just, even though we think of the gastrointestinal system as a tube that's almost external to us, it really isn't. They do live in symbiosis with this. So I think it's fascinating that there's a lot we do with regards to diet that shifts our gut microbiome. And so what do you think with regards to, in your paper, I know you discussed this, the therapeutic potential of dietary interventions to alter our gut microbiome and say, decrease TMAO from a natural perspective?
Yeah. So what you say is really the cornerstone of where the research started. I think Jeff Gordon's work that you quoted, he actually talk about the transmissibility of these phenotypes. One thing that caution this interpretation though is most of us, particularly when we're healthy, has a reasonably robust kind of resident bacteria colonies, should I say. So if you even, try to give a fecal transplant on an otherwise intact,
absolutely,
gut, kind of individual,
yes,
that they actually don't colonize at all.
No, the study I was referring to, I think, was people with C. diff that were obviously
with C. diff. Yeah, exactly,
right.
Exactly. So, and obviously, as a transplant cardiologist myself, just think about we select organs by making sure that they don't have all the bacteria and all that in fecal transplants. It's really, they don't test it at all. They literally so it makes you wonder how little we understand. But I think it has made tremendous contribution. Obviously, to those who are very sick, who have C. difficile infections as a potentially lethal condition, but they live with recurrent, C. difficile colitis, and so it's really an important thing. Now, we have actually, even from a research standpoint, we have actually not shown in an intact gut that you can actually alter TMAO levels. Particularly, there's a study done in Germany that uses donors, fecal donors that are lean and vegan. It does not change TMAO levels, partly in some ways because the original levels are very low. You can't really lower something that's low, So, but that being said, I think the intact, if you have an intact gut microbiome, it's very hard to keep it in. So the only way, obviously, is to alter the environment. So we know from bacterial growth, the change in the environment, just like nature, changes the populations that would favor certain, environmental substrates like food. So if you have food that is, low on carnitine and low on choline, and low on choline is hard because every food, is part of the bilayer of any cell. So you talk about, like, for example, lower amounts of food would actually, reduce TMAO that is otherwise high. And we have actually seen caloric restriction being one of the strategies of lowering TMAO. And in fact, people have actually identified lifestyle modification, particularly with lower caloric intake, actually does lower those who have elevated TMAO levels. We have actually done a study that with Ron Krause at Berkeley, where he actually recruited healthy individuals who have crossover, intake of both white meat, non-wheat, and red meat diets. So the same person at different intervals of weeks would consume the same calorie of food, but the protein composition is different. So what they have found is actually from and this is randomized. It's, of course, it's difficult to be blind, but at least it's randomized in a sequence. And taken together, it was quite clear when and individuals are consuming red meat. Some individuals, not all, had a significant rise in TMAO levels. And when they actually change their diet into non-meat or white meat, it substantially gets lower. And wow. That is actually another kind of illustration and a human illustration that diet in some individuals, those acceptable, are quite vulnerable. Now, I emphasize, not a lot of people interpret it this way. If you actually look, we actually have a graph that shows the top 10% versus the lower 10%. So there are some people that they could eat red meat. Those are the lucky people that actually doesn't have any TMAO go up at all. But others are much more susceptible.
This is so applicable to the way cardiovascular disease clinically is, because I feel like, every time I talk to a patient who says, I don't get it. I have a grandmother that ate, chicken skin and red meat and bacon till she was 95 and never had heart disease. It's like there are just people who genetically, I'm sure that there is just some way that they are not susceptible to those kinds of changes, right?
Yeah. But in this case, it may not be the genetics of the human host.
Oh, wow.
It may be the genetics of the bacteria.
Wow.
It may be the type of bacteria that's in there. And that's what we realize, that what we are dealing with now the human host is one of many things, many factors. There's a lot of factors that are living inside us that we've never, truly appreciate the importance and the impact. In fact, one can argue that this is one of the biggest endocrine organ that we have that is directly related to how we interact with what we do every day, which is eating.
Wow. I mean, This is mind-blowing. This is just unbelievable.
Yeah. And I think for patients, it is important for people to recognize that this is the part that we can control.
Right.
And this is the part that people can modify. And you and I know as clinicians that that's also the hardest part for people to change. Behavior is always hard. We could tell people you got this disease and give this pill. But at the end, if it is just, kind of an effort that they have to make, it is a very difficult thing to overcome. So I think having an illustration like, cholesterol, we took us a long time to actually identify cholesterol from the 70s that it leads to heart disease, to the 4S study, when I was in training that actually showed a significant reduction in events. Then people start to believe it. And then, of course, more recently, with all the new drugs that we can actually lower to the point that one could even eliminate conditions. Dr. Esselstyn, of course, with his, famous kind of strategy for, a plant-based diet that you can really lower cholesterol to a certain level with or without medicines. Yeah. So.
Absolutely. I think this is also fascinating because the previous to your episode, I had a gastroenterologist on who discussed the gut microbiome from the GI perspective. And it's so fascinating, this intersection between cardiovascular disease and gastroenterology. She was at a gut microbiome, the National Gut Microbiome Conference in Florida recently. And she said there's a lot of discussion and research in the GI space. I'm not sure if you can comment on this with regards to finding antibiotics to target things like TMAO, and or finding certain treatments for it. Although we know, obviously, that the goal would be to modify it through diet, but it's just interesting that now in certain areas of gastroenterology, they're actually looking, should this be something that's targeted with medications too?
Yeah, I think obviously our infectious disease colleagues have taught us that is, without knowing what the consequence is actually quite difficult. And certainly we all need a certain amount of all the, with elimination of gut microbes, you also eliminate all the good things that they do.
Absolutely.
And so obviously that is something that we don't have to, if you don't know, don't try, That's a thing that we had to learn more before we could figure out. Now, to be very honest, there have been strategies that our gastrointestinal specialist colleagues have done in very similar manner in, say, irritable bowel syndrome.
Right.
They have used low-grade, antibiotic regimen to reduce the symptom level, and certainly, that is kind of the level one, the lowest level of altering gut microbes. And I personally believe a lot of the drugs, whether it's GI related or not GI related, this whole field of pharmacomicrobiome is actually quite active, meaning that all the medicines and even all the medicinal foods that we have, there's certainly some component of interaction within our gut microbes. And I think that may explain the variability of our responses, but also the variability of the effects. And I think this is the anthropology of medicine now. We're talking about how different cultures have evolved, different learnings of what different foods may impact. As the Chinese, obviously, Chinese medicine is all through the mouth, and in different sequence, and also in different rotations or cycles. And so that certainly how that impacts on gut microbe is something that obviously would be fascinating to explore now that we had all the tools to do that.
It's fascinating. It's interesting to me that the number of genes and all the microbes in a person's gut microbiome is 200 times the number of genes in the human genome. And the gut microbiome can weigh as much as 5 pounds. We're dealing with these bacteria that do, like you mentioned, they do so much for us, but at the same time can also cause a lot of dysbiosis if our guts optimally. So what do you think the most optimal, way, if someone's listening and they want to optimize their nutrition to give themselves that a healthy gut microbiome, what are some of your tips?
Well, I think, as I mentioned, the biggest tip is don't eat too much. I certainly had a pet peeve when I see somebody that lived to 90 plus years old. I always ask them, what are their secrets? And
Me too.
Invariably, invariably they would say two things. Number one is they are avid exercisers. They sometimes kind of, particularly during that era, they may have to walk a lot because it's, people who live to that age obviously had gone through Great Depression. And I think another thing is they have a habit of eating much smaller portions, again, because of the origins of the fact that they don't have a lot to eat when they were young. And so I think, that is a very big component, in my opinion. The second thing, obviously, is to have a balanced diet. And I think, your audience would have, a preponderance to plant-based and, other vegetable, based diets. And obviously, fiber content is also a very important part. We have known for a long time with the short-chain fatty acids, the acetate butyrate and propionate are the ones to actually, those we do have receptors. We actually identify receptors that potentially, the olfactory receptors, that potentially have some bearing in different, important functions in the body, particularly as it relates to blood pressure control. And we've known for a long time, metabolically, they also have very favorable effects. So, in fact, I think it is difficult to study this in the traditional reductionist approach. I think when we start to talk about more of a holistic way of understanding kind of the balanced diet, but also, fresh food, I would caution, based on our research, that many of the supplements may have different effects, even though it's the same compound. It's really the form and the way that we eat them that may actually impact how the bacteria, process them. There's still a lot to learn and a lot to characterize and a lot to understand what is in our food now.
Right.
As a heart failure doctor, we talk about salt a lot, and even the same type of food that you buy 10 years ago versus now, there's actually increased amount of salt. And the processing part we really don't understand. And so also there are many additive substances. For example, some of the food, particularly with the energy foods and certain, kind of protein building foods, have carnitine on it. Then these carnitine, obviously, if we are doing experiments that identify carnitine as being a potentially, create the opportunity for these bacteria to make TMA, right, it certainly is a risk if we actually unknowingly taking in some of the food that has such additives. And so this is something that, as a public health, importance, we need to learn what we are putting in our mouths.
That is so well said. It is absolutely brilliant. I have a question for you, a clinical question for you that I wanted to ask you for my own practice. What do you think the utility is for us cardiologists, general cardiologists that do a lot of work in prevention, testing for TMAO? Do you think that it currently should only be used in the research space, or do you think that this is something that's going to start integrating itself into our outpatient tests as well?
Well, I think the clinical practice, as you know, always gears towards the demonstration with evidence of incremental benefit.
Right.
And so I think most biomarker, and my background actually is in heart failure biomarkers, so I could also translate that into what TMAO is going through. And I actually have no conflict of interest in that. What is important is the biomarker, we need to understand its origin, its natural history, and how it provides incremental insights. And so take, for example, when we test a heart biomarker called BNP, and I happen to, in the very early stage of my career when BNP was clinically available, and even up to now, we have randomized controlled trial that is ambivalent in terms of trying to understand how routine measurement is beneficial, but at the same time,
but I order it all the time.
Useful, exactly. When we measure individual patients, this is actually one of the most consistent tracker of badness. This is something that is very important for us to better understand the disease severity. In fact, the ASA/CHA guidelines actually said that, for people, for prognostic value and for diagnostic value, when they admitted to the hospital, it's a class 1, for diagnostic. And even there's more about, population-based studies that show, at least now the guidelines actually, describe. In fact, the European guideline just came out this week on the use of natriuretic peptides. It is something that, ironically, I always think that if we didn't have echo later than BNP, we would have doing all BNPs and only some people would do echoes. That being said, TMAO came almost as, again, firstly, it's something that people are not familiar with. We're still learning a lot about the natural history and the various scenario and how it's tracking with certain disease states and how it may be informing care. I do think that for some individuals, particularly when they have strong family history or premature coronary disease, as a guidance of therapy, particularly with behavioral, modification or dietary modification that needed a lot more, convincing. And this is something that is helpful. And clinically, it is available, although only in certain places. I think it is something that we would learn over time as to whether this is potentially provide, a true clinical insight. Although, as you know, even many of these novel biomarkers in the preventive cardiology world still need to get to the higher bar, which is that we need some, true, consistent studies to show there is an incremental benefit. And we had this discussion, obviously, with even the most common biomarkers like CRP, and like, and it's always kind of the believers would feel that it's good. And even for CRP, there is a Jupiter, and there are studies suggesting statin is helpful. And even LDL, there was a whole argument about, whether you need LDL for, cholesterol management, So I think it really comes down to our community as to what we believe is the most important aspect and how does biomarkers like TMAO can help with the management. I think the essence of this, however, is to really shed a light on really the physiology that we may have overlooked all through the years until, more recently, that we are dealing with not just, a one pathway, but an entire, environmental change that is based on lifestyle. And I think this is where that conversation needs to be important. A lot of people like to ask, what's good food, what's bad food? It's very difficult because it really depends on what is inside you as well.
I mean, you couldn't have said it better. That's absolutely outstanding because you're right. I mean, you've added the research you've done and the research in this space of the gut microbiome has added an entirely dynamic and complex layer into the way we look at patients. Because now we're not just looking at the patients and their family history and their genetic makeup. We're thinking of these millions upon millions of bacteria that live in their bodies as well. And so it really does give an opportunity for us to look deeper into more ways that, and also really how our organ systems are all related. I think it's fascinating too, because, there's a reason why we see people with autoimmune disease who are also at higher risk for cardiovascular disease and so on and so forth. So it really is more layers to the story.
Yeah, I think the population that we will expect to have more insights probably will be those that definitely had the propensity to accumulate TMAO, which will be the patients who have renal insufficiency. And we already know there's a lot of dietary discussion as to how to advise, true renal nutrition and how to modify. And that's where I think a lot of advances would occur. Diabetes is always a fascinating area and an area that has not just the gut microbe, but also metabolic alteration with obesity and with different pathways. And I would like to emphasize that TMAO is one of probably, thousands of pathways and that we just don't even recognize at this point. So stay tuned. There will be a lot more to come, but a lot more mechanistic studies are also needed to demonstrate that indeed there's something there rather than just, somebody tells you that, okay, trillions of bacteria is good for you, so take it every day. We don't know really until we actually understand more.
Absolutely. I can't thank you enough for doing this. And for all of the research and work you've done is just absolutely outstanding. So thank you so much, Dr. Tang.
Thank you. And I want to definitely credit for where credit is due, Dr. Stan Hazen's lab and all the postdocs and all the folks that actually work in this area, and also all the collaborators too. And of course, the National Institute of Health in terms of their funding.